Hi, everyone. Thanks so much for coming in the middle of your workday. I'm really excited to be here. My name is Emily Anthes, and I'm a science journalist. And I'm here to tell you a little bit about my new book, which is called Frankenstein's Cat: Cuddling Up to Biotech's Brave New Beasts. The idea for this book came to me gradually over the course of a couple of years. I'm a big science geek, and writing about science is what I do for a living, so I spend a lot of time reading the science news and the journals and press releases. And over the period of several years, I started noticing more and more of these strange headlines. I would see things like, "Scientists produce glow-in-the-dark cats," "Synthetic biology and the rise of the spider goats," "Robo-rat controlled by brain electrodes," and on and on and on these headlines seem to go. These headlines, and many others like them, made it clear to me that science had given us this whole new toolbox for tinkering with life. We were reshaping animal bodies in profound new ways, altering their genetic codes, rebuilding their broken bodies, and supplementing their natural senses. So each of these stories was interesting on their own, but I began to wonder about the bigger picture. We had all these new tools now. How and why were we using them? And what did it mean for animals? And what did it say about us as humans that we were making these sorts of changes? So I don't have time to answer all of these questions in detail today, but I thought what I would do is at least give you a glimpse into some of the things that are now becoming possible. One of the things I learned in researching this book is that lots of the developments and ideas that have traditionally been relegated to science fiction are now, in fact, quickly becoming reality. So obviously, when you talk about animal modification, one of the first things that comes to mind is genetic engineering. And my adventure into the world of genetic engineering actually started not far from here. It started at the Petco over at Union Square. And I went there because I wanted to buy myself a collection of glow-in-the-dark pets. It's not as crazy an idea as it might at first sound. Scientists, for instance, have known for many years that there are some creatures out there, particularly marine organisms, that happen to glow naturally. One of the most famous is this jellyfish known as the crystal jellyfish, and it's famous for naturally producing what's known as green fluorescent protein, or GFP. This protein absorbs blue light and re-emits it as green, yielding the green fluorescent ring you see around the jellyfish's body. In the 1990s, scientists managed to isolate the gene that codes for GFP. And after they did this, they realized they could start moving it around the animal kingdom, take this GFP gene that comes from a jellyfish and put it into all sorts of other critters. That leads you to animals like this. This is a monkey, and only one genetic tweak has been made to him. All that scientists have done is taken this GFP gene and put it in his genome. As a result, his body naturally churns out these green fluorescent proteins, and under a black light, like what you see here, he glows in this eerie way. This photo has not been altered at all. So this kind of technology is incredibly useful to biologists because not only can they make whole animals glow, they can engineer animals that glow only in certain cells or only in certain tissues or only during certain phases of development. And so by creating these animals, they get insight into biological processes and what's going on inside the body of these animals. So from a scientific perspective, it's an incredibly useful technology. But about 10 years ago, some young entrepreneurs thought that glowing animals were kind of cool, and maybe there'd be a market for using this technology to create neon pets. They started with this fish, known as the zebrafish. It's an incredibly common aquarium fish. You'll see them over at Petco, most pet stores. It's a very small tropical fish. It normally has these black and white stripes, which is what gives it its name of zebrafish. And it's been in use in the ornamental fish industry for many, many decades. These entrepreneurs decided to start with these zebrafish and then to transfer fluorescence genes into zebrafish embryos. Not just green fluorescent protein, but red, orange, purple, all of the colors of the rainbow. When you do this, you turn zebrafish into these. These are glowfish. They are America's first genetically modified pets. They have been on the market since 2004, and they're now widely available. They're sold at most Petco's, most PetSmarts, Walmarts. They also come with a range of accessories, so you can buy a special glowfish kit that comes equipped with, it's an aquarium that comes with blue and black lights built in, so when you turn these lights on, it really brings out the glow in these fish. So scientifically, these fish are not very radical. Moving fluorescence genes around is one of the simplest kinds of genetic engineering that scientists do, and it's very, very common in the lab. But what's remarkable about them, and why it was the first thing I did as I researched this book, is it's really the first biotech animal product that is widely available to the public. They cost $5 or $6 a fish, so anyone who wants to can just walk into Petco or Walmart and buy themselves a whole aquarium full of these high-tech, next-generation pets. Of course, this is a trivial use of biotechnology, and the company that makes these fish has been criticized for using biotech for frivolous purposes. But once you've developed the technology, it can be used in all sorts of ways. There's a lab in Singapore where the scientist is doing the same thing. He is transferring fluorescence genes into zebrafish. But he's not creating pets. What he is trying to do is create living pollution detectors. He's managed to couple these fluorescence genes with genetic switches. So what happens is if these fish swim in water that's contaminated with certain pollutants, they suddenly begin to glow. They switch from black and white to these neon fish. So that's one example of how you can take this GFP technology and use it in a very applied way. Over the course of my adventures, I met some other extremely useful genetically modified animals. I went out to UC Davis in California and met a herd of genetically modified goats. They don't look as interesting or strange as the fish, but they are genetically modified on the inside. And these goats have been engineered to produce high levels of a compound known as lysozyme in their milk. Lysozyme is a naturally occurring enzyme, and it has antimicrobial properties. What it does is burst bacterial cells like balloons, essentially, and can be used to combat E. coli, salmonella, other sorts of diseases. The idea is that the milk from these goats, which is high in lysozyme, could perhaps be fed to children. Diarrheal disease is not a huge problem in the US. It's not something we normally think of as a serious public health concern, but in many places around the world, it's a major cause of childhood mortality. So it's not in human trials yet, it's still in animal trials, but so far this milk does show some ability to promote intestinal health and help piglets fight off various forms of diarrheal disease. The goal would be eventually to take this milk into human trials and perhaps to get these genetically modified goats out in the fields producing milk that anyone can drink. Of course, you can't talk about animal biotechnology without talking about cloning. That's often comes up right away. And as it turns out, a university in Texas, Texas A&M, happens to have become a leader in animal cloning. They're an amazing university. They've cloned 6 different species of animals so far, and they're working on cloning more. And so I went out to meet some of these clones and talk to the scientists there. While I was there, as an unexpected bonus, I had the chance to meet the world's first cloned cat. She was created as part of a project that began in 2000 when a billionaire partnered with some scientists at A&M because he wanted to clone his beloved dog. When the scientists were talking to him and working out the details of this project, they realized that there might be a market for cloned pets that went beyond this billionaire that many Americans might want to clone their pets. So they decided that together they would launch a company that would offer commercial pet cloning services. They called this company Genetic Savings and Clone. One of my other discoveries is that animal cloners love puns and love wordplay, and so it is all over the field. This is just one of many examples. But when they launched the company, cats and dogs hadn't actually been cloned yet. So before they started selling their services, they realized they would need to really nail down the technology and make sure that it worked. What they decided to do first was try and clone a cat named Rainbow. This is Rainbow. And the word cloning gets thrown around a lot. You see it in headlines and in short news articles, but I find that there's not a lot of discussion about what cloning actually means and how it works. So this talk isn't too technical, but if you bear with me for a minute, I'd like to walk you through the process of how the scientists went about trying to clone Rainbow. They started by harvesting some of Rainbow's cells, but the trick is you can't just take a random cat cell, like a skin cell from Rainbow, and pop it into a new cat uterus and expect a whole new cat to grow. Biology doesn't work that way. So you need to get the DNA from Rainbow's cells into the proper vehicle, which is an egg. So after they harvested cells from Rainbow, they recruited, euphemistically, some feline egg donors. They harvested unfertilized eggs from all sorts of other female cats. Then they used a tiny pipette, which is like a small little turkey baster, to essentially suck out the nucleus of all of these eggs. So you might remember from high school biology, the nucleus is what contains the chromosomes and all the genetic instructions. So basically what they're doing is taking the DNA from these eggs out and leaving you with an empty egg. Then they took one of Rainbow's cells and injected the entire cell into the empty egg. They put it just in between the inner and the outer membranes. Then they applied an electric current to this whole cell within a cell system. What the electricity does is basically turn the cell membranes into Swiss cheese, poking holes in the membranes of both the egg and Rainbow's skin cell. As a result, the DNA from Rainbow's cell can flow into the empty egg. This sudden influx of genetic material actually tricks the egg. It thinks it's just been fertilized by a sperm, and it will then go on to grow and divide just like a normal embryo. What you end up with then is a cloned embryo, an embryo that has all the genetic material from Rainbow herself. These embryos are transferred into surrogate mothers, other cats that have been recruited to the project, and if all goes well, after a normal period of gestation, these surrogate mothers give birth to a clone. Things did in fact go well with the project to clone Rainbow, and scientists ended up with a healthy little cat clone. This is her. She doesn't look very monstrous at all. And her name is actually CC, which stands for carbon copy, and also copycat. So again, scientists having fun with naming. And scientists did various tests on her. They found she was, in fact, a clone of Rainbow, and that she was healthy and seemed to develop normally. After her service to the laboratory was done, one of the scientists involved in creating her, a man named Dr. Dwayne Kramer, adopted her. So when I went out to Texas, he invited me over to his house to visit Cece, who was about 10 years old at that point, and I eagerly took him up on his offer. The day did not exactly unfold as planned, though. When I got over there, I naturally started to approach the front door of the house, figured we'd be going inside to meet his cloned cat. But Dr. Kramer and his wife came running out and sort of said, "Oh, no. Cece has her own house for her and her husband and their kids," was what he said to me. And so he sort of steered me around to behind the house where the humans lived, and sure enough, in the backyard was this 2-story house. We're not talking about a little dog house here. This is a house that's bigger than my Brooklyn apartment. And so he opened the door and let me in, and there were 5 cats running around inside. So after they'd adopted Cece, they thought she might be lonely, and they adopted a male cat named Smokey to keep her company. One of the things we do know about clones is that they can reproduce normally, and Cece and Smokey did go on to breed and had 3 kittens conceived in the usual way. Here are a couple photos I took from inside the cat house. In the first one here, this is Dr. Kramer, one of the cloners, trying to give Cece a pet. She was not very cooperative the day I was visiting. She spent a lot of time sort of sitting out in the window looking over what she surely imagines to be her kingdom. She's a very pampered cat. There are ladders or steps here to the second floor of the cat house. Dr. Kramer also keeps all of his students' dissertations up there, so it's a combination library cat residence. And they also build this outside screened-in area for the cats, so they can go out, get some fresh air. They don't have to worry about the cats running off or being attacked by some sort of predator. So the cat family is still living out in Texas. They all seem happy and healthy so far. And Genetic Savings and Clone did go on to clone a couple cats for regular American customers. They cloned 8, I believe. But the price was high and the demand was kind of low, so the company eventually shut down. Again, you can think of pet cloning as a pretty trivial use of biotechnology, and it is. But the advances that let Cece be cloned paved the way for other applications. This is Ditto. He is the world's first cloned African wildcat. And African wildcats are a species of small, exotic cats, and they're threatened in the wild by habitat loss and also by interbreeding with domestic cats. His name is Ditto, which is another great pun, I guess, by the scientists. And he was created by scientists in New Orleans who work at what's known as the Audubon Center for Research of Endangered Species, or ACRES. The ACRES scientists have really made a name for themselves by using all sorts of reproductive technology to help endangered species breed. They started with IVF and created test tube gorillas and things like that. But as the technology has advanced, so has their work. And in recent years, they've become especially known for their cloning projects and their cloning of small species of endangered cats. They've done 4 or 5 of them now. So they've just done small numbers of these cats, but the idea is that perhaps if we can refine this cloning technology and really get it to work well and predictably, that we can use cloning to help prop up species that might be struggling in the wild. There's also Bruce. This is another animal clone. He's a cloned bull and was also created at Texas A&M. Bruce is the clone of a special bull named Bull 86, who was discovered in the early 1980s. What was amazing about Bull 86 is that he had a natural genetic mutation that just happened to make him resistant to all sorts of livestock diseases. Because of this one genetic error, he was resistant to tuberculosis, salmonella, and a disease known as brucellosis, which is a contagious livestock disease. Unfortunately, Bull 86 got old and died before the scientists could breed him, and though they saved some of his sperm, accidents happen, as they can in science, and it was accidentally destroyed several years later. So for a while, it seemed like Bull 86's valuable genes had died out with him. But cloning came along in 1996, that's when Dolly was born, and the scientists at A&M suddenly had an idea. Stashed away in the very back of the freezer was a skin biopsy that they had taken from Bull 86's ear 15 years earlier. They took the biopsy from the freezer and used the genes it contained to clone Bull 86. They ended up with this bull here. Technically, his name is Bull 86 squared, which in their scientific papers, the scientists say stands for his, quote, exponential genetic potential. But because he was also resistant to the same diseases, TB, salmonella, and brucellosis, students took to calling him Bruce. So this is just one animal, one bull that's been cloned, but his birth raises intriguing agricultural possibilities. Lurking out there in our herds and flocks might be other animals that, unbeknownst to us, happen to have natural genetic mutations that protect them from disease. So one possibility is that if we can find and identify these animals, we can use cloning to create sort of new stocks of breeder animals and breed some of these helpful disease-resistant genes into larger livestock populations. So it's not just genetics. We're also remaking animal bodies from the outside in. And in particular, advances in veterinary medicine and materials science are letting us give animals state-of-the-art prosthetic limbs. One of the leaders in this field is a British veterinary surgeon named Noel Fitzpatrick, and he is sometimes known as the Bionic Vet. He was the subject of a documentary series by that title, and that's because he's become best known for his work giving people's pets bionic limbs, prosthetics that are permanently implanted into their bodies. The device he invented is called the intraosseous transcutaneous amputation prosthesis, or iTap, as everyone calls it. And that's kind of a mouthful, but if you break down what those words mean, you really get a good sense of what it does. So osseous comes from the word for bone. Intraosseous means inside the bone. Cutaneous from the root for skin. Transcutaneous means across the skin. And then, of course, amputation and prosthesis. So if you put this together, what you end up with is a prosthetic device that gets inserted inside the bone and crosses through the skin. This on the left here is what the iTap actually looks like. It's made of a titanium alloy. That's what this rod is here. And this longer portion here gets implanted actually inside of an animal's bone. This is an X-ray of a dog who had the bottom portion of his leg amputated. This smaller metal piece here then juts out through the skin and sort of into the open air. The idea is that once this implant has been surgically implanted and the animal recovers, you can then pop different artificial paws onto and off of this little metal peg here. You'll also notice that there's this sort of umbrella-shaped cap, and it's a little hard to see, but that is also implanted just underneath the surface of the skin. It's covered in all of these little tiny holes, and that's a crucial part of the iTap device. These holes allow the device to make a sturdy, permanent interface with the body. What happens is the soft tissues, the skin, the collagen, the muscle, actually grows into and through these holes, creating this permanent link between implant and flesh. One of the first animals to get one of these iTap devices is a bulldog named Cole. When Cole was 8 years old, he developed a tumor in his front left paw. And the traditional treatment for these kinds of bone tumors is often just to amputate the paw and to let the dog go on with its life. We've probably all seen three-legged dogs, and a lot of them get along just fine. But not all dogs do well on three legs, and veterinarians were particularly worried about Cole because he had severe arthritis. And so they worried that if they took away one of his legs, his remaining three legs might not be strong enough to support his fairly massive body weight. So they agreed to give him an iTap. This is Cole. A few weeks after the surgery, he had the device that we were just looking at implanted. You can see the metal peg sticking out here, and the rest of the iTap is sort of hidden away inside his leg there. Once he healed and recovered from the stress of the surgery, then his owners and veterinarians could experiment with different kinds of artificial feet. This part of the iTap is still sort of a work in progress. Engineers and veterinarians are working together to figure out what designs of paws work best and in what scenarios. Here's a wide variety of the designs that have been tried. And in Cole's case, they found that what worked best was actually one of the very low-tech solutions. This last one here, it's essentially just like a rubber sink stopper. So they popped this onto the metal peg that came out of Cole's foot, and this is what they got. This is Cole after he's recovered from all of his surgeries and all the procedures with his new prosthetic paw. It was a huge success. He tolerated the implant well. It did, in fact, grow into his skin and become a permanent part of his body. And he seemed to go back to life as a normal dog. His owners reported that most people never even noticed he had a prosthetic paw unless they looked closely. And also, before Cole had gone through all these medical procedures, he had been taught some of those conventional dog tricks, where you can say paw and the dog gives you his paw. And after all these procedures, Cole would in fact do it with either his normal paw or his prosthetic paw. So he seemed to get right back to life as a dog. Fitzpatrick has also done cats. This is Oscar, and he has the distinction of being the first recipient of the double iTap. You can see both of his back legs have been replaced here. Oscar, unfortunately, had a run-in with a mechanical grain harvester and lost both of his back paws, but they gave him 2 iTaps. It's the same general procedure that we saw with Cole, just at a smaller scale. I've got a short video I'll show you here where you can see Oscar walking around on these paws, and Fitzpatrick also explains a little bit more about the design.
Now been 3 weeks since we put on Oscar's new feet. He likes his new blades, don't you, Oscar? Good boy. He also likes fish. But he can run and jump on them now, and his feet are moving very well indeed. Come on then. Come on, bud. So you will have seen his prototype feet, which were just compressive devices. They were devices which just had a little spring in them. And that was just to encourage him to use his feet in the first instance and to remodel his bone so that it built up around the metal. Now he's moved on to a different phase in his life where he needs to be able to run. We can see that his new foot is like a blade. It hits the ground like a normal foot and has a little sole on it so he can get some traction and works like a seesaw. The foot was designed so that the metal inside his bone couldn't break. So if Oscar gets into difficulty, the foot will break here at a break point before it would break the implant inside his bone. It's very much like the bindings on a pair of skis. So it will break off here at the binding before it breaks your leg, which is the way skis work. And we designed this implant with that in mind. So if you're skiing down the hill and you catch your foot, it will snap here before it breaks your leg. So that's pretty cool.
So Fitzpatrick has now done about 24 of these animal iTaps, and one of the amazing things about them is that they've been so successful that the iTap is now in human trials in Europe. One of the first patients is a woman named Kira Mason, and she lost her arm in the bombings of the London Underground in 2005. She had tried a variety of different prosthetic devices and found them lacking, and so she signed up for this clinical trial and got basically the same sort of iTap that Cole or Oscar got. She reports that this new iTap arm is much more convenient and comfortable for her to use. Finally, towards the end of the book, I get into sort of the most extreme territory, and I start talking about animal cyborgs, of which there are now a variety. In particular, DARPA, the research and development arm of the Pentagon, has been interested in developing remote-controlled, steerable insect cyborgs. And this is because DARPA has long had an interest in creating little tiny flying drones that are much smaller than the ones we tend to hear about today. And the idea is that these tiny drones would maybe fly into occupied buildings or distant caves and perhaps conduct surveillance, send video or audio feeds back to military personnel and let us sort of scope out a territory before we actually go there. The difficulty is that it's hard to build these completely mechanical drones at a very small scale. Partly that's because the dynamics of flight change when you get that small. And there's also a battery problem. These drones are so small and light that they can barely carry any weight. And as a result, the batteries they do carry tend to be very low power. They only keep these drones aloft for a couple of minutes. So DARPA realized that maybe insects would be a shortcut to these kinds of drones. After all, they already know how to fly, and better yet, they power themselves. All we'd need to figure out how to do is to steer them and make them obey our commands. So in 2006, DARPA put out a call asking researchers across the country to submit proposals for how they would create these kinds of insect cyborgs. DARPA funded a couple of these proposals. One of the scientists who won funding is a man named Michel Maharbiz, and he's at UC Berkeley. He decided he was going to work with these flower beetles. They're relatively large beetles, 3 or 4 inches, so they're bigger than most insects. The trouble is that Maharbiz is an electrical engineer, and he knew plenty about the wiring and the circuitry, but next to nothing about insect biology. So to begin with, he started by delving into the literature and trying to figure out what we knew about how these things fly. He ended up homing in on 2 areas of the beetle brain at the base of each optic lobe, here and here. And the literature suggested that these areas of the brain were involved in flight initiation and cessation. So he tinkered around in his lab and decided he was going to hack into these brain regions. He ended up threading thin wires into the base of each optic lobe. The wires then snaked out of the brain and connected to the circuit board. Then at the back of the circuit board here, you see a big radio antenna. The scientists wrote specialty software, which they called the Beetle Commander, and they could sit with their laptops across the room, 100 or more yards away from the beetle, and start issuing commands with this software. The commands would be picked up by the radio antenna, transmitted into the circuit board, and then electrical pulses would go zinging down these wires and into the beetle's brain. Through trial and error, they discovered that if they sent a series of rapid-fire electrical pulses into the beetle's brain, it would suddenly take flight. On the other hand, if they sent a single long pulse to the same area of the brain, the beetle would immediately stop flying. The effect was so dramatic that a beetle would literally fall out of the air in mid-flight. And you don't have to take my word for that, because you will see that in this video. Were not injured. So that's pretty dramatic, but of course that's just step one. In order to actually be useful, the beetles need to not just stop and start, but go in the right direction. And so the next step was to figure out how to take control of the beetle's movements while it was in the air. The scientists managed to do this by adding a second set of wires. It's not in this photo, unfortunately, but they went to the base of the beetle's wings on each side. And what they discovered is that when they stimulated the right wing muscle, the right wing would start beating with more force, and as a result, the beetle would naturally veer to the left, and vice versa. By stimulating the left wing muscle, the beetle would veer to the right. I have another video of that. I'll warn you in advance, it's a little hard to see than the previous video, but you can see definitely the beetle veering across the room. So I think I'll stop it there. You guys get the idea. It's a little difficult to see. But this is a pretty dramatic thing to do, to take control of these beetles' movements, but it's still pretty crude. So right now, all we're able to do is make them sort of veer in one direction or the other. In order to really be useful, we'll need to take much more precise control. You know, military commanders may want the beetle to go left by 40 degrees while flying at a certain altitude and speed. So that's something that the scientists are working on now, is to make much more fine-tuned commands that they can control the beetle with. So obviously, some of these things are very radical things to do to animals, particularly when you talk about taking over their nervous systems. And it's easy to understand why some of these technologies are controversial. It can certainly, in certain cases, cause animal suffering. It can turn animals into commodities or tools. And in certain cases, it could potentially pose a risk to us or to the environment, to ecosystems that we all share. And these are absolutely serious issues, and they deserve serious consideration. There is plenty of reason to be cautious as we move ahead with these technologies. I think we need more of a societal discussion about how we want to use these technologies and why and to what end. And we should absolutely be prepared to reject applications or products that pose undue dangers. But I also think that there's a lot of reason for hope. And animal biotechnology could do more good, not just for the world, but also for animals themselves than it's often given credit for. Biotechnology, after all, is not inherently good or bad. It's merely a set of techniques, and we have choices about how we can use them. What scares me a little bit is that if we have these knee-jerk reactions against biotechnology, then we'll lose many of the good beneficial applications along with the bad. So thank you guys for having me. I know we don't have a lot of time left, but I'm happy to take questions. Yes?
I hear there are glowing plants on Kickstarter. Have you seen them? What is your sentiments about them?
I have seen them. So the question, there's a Kickstarter campaign to fund the creation of glowing plants, and it's actually, that's not a particularly radical thing to do. So scientists have created glowing plants in the lab, but one of the things that's become controversial about this project is that one of the rewards for donating is a set of these seeds, supposedly, these glowing plant seeds. So a big concern with genetically modified organisms is what happens if they get loose in the wild. And so I think the plants are likely to be completely harmless. I'm not worried about human safety or, if someone ate them, food safety. But I think it's a little bit worrisome that we might see all these seeds going out there in a totally unregulated way. So I think that's why it's gotten so much attention. Yes?
Just wondering, with the cloning to save animals that are dying out, if every animal is a clone, how would the population not end up just being interbred? Like, is that something that...
Right. So cloning is not a cure-all, and sometimes it's presented as, well, all we have to do is make more copies of these animals, problem solved. I think that's an exaggeration, but I think it can be useful in much more limited ways. So as you, I think, we're getting at, one of the big problems with endangered species isn't just that they are small in number, but they lack genetic diversity. And so cloning obviously doesn't solve that problem because it only gives you replicas of the animals you already have. But it could potentially be used to keep diversity from dwindling further. So if you have animals that die before they can reproduce or are infertile for some reason or something else happens, you could potentially take cell samples and clone them and at least sort of get those genes back into the population. There's also a big effort right now with frozen zoos. So, like tanks of liquid nitrogen where scientists are storing cells and DNA from all sorts of wild animals. And I think there's real potential there because if we have a species that, say, now looks okay, but in 50 years is endangered and the genetic diversity has died off, we may have these samples of genes that have disappeared and can perhaps clone them back into existence. So I think in this limited way, it could be useful. But yes, we're not going to save species just by making thousands of clones. Yes?
On a related topic, I saw on 60 Minutes, maybe a year ago, that they found some dinosaur DNA, and they were actually, for real, legitimately trying to actually bring some of them back. What's your perspective on that, or do you know anything about it?
I haven't seen that. The perspective of every scientist I've ever seen is that dinosaurs will never be possible. Not only cells degrade over time, but DNA begins to degrade at the moment of death. And so I've not seen anyone say that the DNA is just too far gone. The main thing that there's the moonshot project right now is the mammoth, which is a lot more recent than the dinosaur, and it's still unclear whether we will have good enough DNA. Even these best preserved samples, and you probably saw just last week, I think they've found an amazing preserved samples, but so far the DNA is still in fragments, the cells are deteriorated. So, scientists are still hoping, but it's, to excuse a mammoth project, it will take not just taking a cell and popping it out and cloning it, but we'll have to painstakingly reconstruct what we think the mammoth genome looked like. And so even that's a bit of a stretch, but dinosaurs really seem like an impossibility. Yes?
For the mammoth project, though, what species would we use as a host?
The discussion is the elephant, which, well, we don't know. And that's, I don't think we will know. And, there are also questions, I think, about whether, even if we can do it, it's fair to do it. At least my thought is that the ecosystems for these animals have disappeared, and the Earth is so radically changed that what would we do with these mammoths when we brought them back? Would they just live in zoos? Would it just be for our own amusement? Just personally, my perspective is I would rather see this technology used to try and keep the species we have now from dying out rather than trying to bring back species that have been gone for a long time. But others disagree with me. Yes?
You said one of the reasons the genetic saving and clone went out of business is because it was too expensive. Is the cost coming down for technology?
The cost of pet cloning has not come down a lot. So right now there are two commercial, I don't know of any commercial cat cloning companies in existence, but there are two dog cloning companies in South Korea, and it's $100,000 to $150,000 still to clone a dog. But there are some companies in the US that are banking on it coming down, and they actually will store your pet's DNA, and their whole business model is when cloning gets better and cheaper, then you can clone your pets. We'll see, I guess. Do you have a follow-up?
I think I saw that CC was different colors than her clone. Is that a disappointment for pet owners to realize it's not exactly the same?
Yeah, so I talk about this in the book, I didn't get into it here, but if you were watching closely, you'll notice that CC was gray and white and Rainbow was gray and white and orange. And it turns out that calico cats in particular are not great candidates for cloning because the genes for sort of gray versus black fur are on one X chromosome, and the gene for orange fur on another X chromosome. So in calico cats, depending on which X is active in a cell, you either get a patch of black or orange. But when you clone, scientists suspect that what happens is the cell they used from CC had the X chromosome was turned off in that particular cell, and as a result, CC didn't express any orange. The scientists were actually, the entrepreneurs were not happy about this because they thought it would, people would realize that it might not be a twin. The scientists didn't do this intentionally, but were really happy to see that CC didn't look the same. One of their concerns has been that pet owners might misunderstand the technology and think they're getting, their same old pet back. So for them, they use this example a lot to remind people, like, it's not the same animal. But calico cats, you're always going to run that risk in particular. Yes?
What effect does the mitochondrial DNA have on this whole process?
So that's the other thing that you don't hear talked about very much in cloning. So remember I showed you when they take the egg and they suck the nucleus out, that's 99.9% of the genetic material. But mitochondria, which are these little organelles that sit in the cytoplasm, have their own tiny little genomes. And so technically, clones aren't quite perfect copies of their genetic donors because they have the mitochondrial DNA from their egg donors. Most scientists sort of wave that off. Because it's so specialized and small, they tend to not, maybe it will have a slightly different, energy metabolism, but because it tends to not have physical, visible effects, scientists don't really talk about it. But, it is a small, complicating factor.
That would definitely make ancestral analysis confusing.
Yes. Well, and one of the issues with cloning endangered species is because, like, if you have a leopard and you're trying to clone it, you don't want to put all these other, like, endangered leopards through surgery to harvest their eggs. So they tend to use eggs from domestic species, like domestic cats. And so there's an interesting philosophical question you can ask about, if it's a jaguar, we're reproducing these endangered species, but it has a tiny little bit of DNA from a different species. Are we really continuing the lineage of the same species? And that's, a debate that ecologists are having, and it's an interesting philosophical question. Yes?
You mentioned that one of the X's was turned off, and that's why the clone looked different.
Mm-hmm.
Is it possible that if we were to use, I'm not sure what cells were used initially, if they were like skin cells or something else, but if we use stem cells, would that make a difference?
It could. One of the problems with a lot of this work is that so few animals of any one species have been created that there's not a lot of data to work with. I don't believe that's something that scientists have tried. The idea is when you clone that all the genes should get, so if you take DNA from a skin cell, it's what's known as differentiated. It's specialized to be skin cell DNA. And in theory, when you put it in the egg, it's supposed to get reset to sort of the embryonic state. But that doesn't always happen, and sometimes it happens partially. And so it's a question of sort of how well we do at getting the DNA to sort of reset to its normal settings. And that's thought to be one of the reasons that clones sometimes have birth defects or die in the womb, is because if the DNA doesn't get reprogrammed correctly, it may not be viable.
So is the other X on anywhere else in the clone? So the clone is female, and there were two X's, and one of them was turned on, the other one was off in the cell that was shot into the embryo.
Right.
So was the other, did they do any tests to see if any other parts of the cat?
They didn't, and X inactivation is sort of their best guess for why she didn't have any orange, was that for whatever, each individual cell, a different X chromosome can be on. And so, in theory, it could have turned out that some of the other X's, the orange X's, were on in some cells, but they didn't really, look into it in detail. Their guess is that for whatever reason, the DNA didn't reset, and so in all the cells, that X chromosome was inactive. But it's not clear. Yes?
A really stupid question, but how does it work with male? Because it sounds like we're using genetic material from a female animal and plugging that with more female. Does cloning actually work in the male instance?
Yeah, it does. But you'll get, if you clone a male cat, you'll get a male cat. If you clone a female cat, you'll get a female cat. And men have mitochondrial DNA. It just comes from their mothers. So, you can clone either sex. Any other questions? Thank you guys for coming. Really fun to talk to you.